Welcome to the Proteomics course. In today's lecture, we will talk about generating protein microarrays, and we will focus more on nucleic acid programmable protein arrays, also known as NAPPA. So, as you know, the microarrays provide high throughput versatile platform for large scale analysis of functional proteins. The traditional cell based expression methods which have been used for protein microarrays, they generate proteins in the heterologous system such as Escherichia coli. But there are several challenges posed by these systems for protein purification as well as maintaining the protein integrity and functionality, protein storage. All these pose several challenges. So, to overcome these obstacles posed by the traditional cell based protein purification methods, the various cell free expression based systems have been generated. In the last lecture, we discussed about cell free expression methods and how different type of protein microarray platforms have been generated by employing cell free expression based systems. So, in the previous lecture, we discussed about various type of cell free expression based protein microarrays. We talked about protein in situ arrays, halo tag arrays, DNA arrays to protein arrays, multiple spotting technique as well as nucleic acid programmable protein arrays. Although each one of these technique offers several unique advantages, but NAPPA approach has proved and overcome various limitations, and it is applied for very high density arrays. So, today I will focus more on NAPPA arrays. Just to refresh you about nucleic acid programmable protein arrays, which we discussed in the previous lecture. As you can see in the slide, you have a microarray slide, and each feature which I have highlighted in the expanded view contains 4 features. One of that is cDNA containing GST tag, then you have bovine serum albumin BSA protein, capture antibody such as anti-GST and BS3 cross-linker. So, in NAPPA approach, plasmid encoding the target proteins are fused with an affinity tag, and those are affixed to the array surface. The array surface is activated by the addition of a cell free expression system, which could be wheat germ expression system or rabbit reticulocyte lysate. Rabbit reticulocyte lysate is more preferred for eukaryotic mammalian protein expression. So, the target proteins are expressed and immobilized in situ, and they are detected by using an universal anti-tag antibody. So, in this slide I have shown you the anti-GST tag and anti-GST antibody. So, if you have GST tag, you can use anti-GST antibody. If you have flag tag, you can use anti-flag antibody. If you have MIC tag, you can use anti-MIC antibody. So, these universal tag strategies can work out depending on what tag you use. You can use corresponding antibodies for the same. So, with little bit overview of cell free expression based systems, different type of protein microarray platforms which we have discussed. Today we will focus more on how to make protein microarrays by using nucleic acid programmable protein array approach. I will walk you through with the workflow step by step how various parameters one need to monitor. I will talk to you about each step, for example, cloning and plasmid preparation, aminosilane coating of the glass slides, what type of printing parameters one need to ensure for good printing quality. Then in the NAPPA approach, one need to ensure that DNA printing is of good quality as well as protein expression is good So, all those quality control checks as well as what different type of control features one need to have on these arrays, so that during various type of application one can make sure that they are relying on the good data. So, we will walk through all this process in the workflow. So, what are the requirements to assemble a protein arrays? So, in today's lecture, when we will discuss about making protein arrays, that is quite similar for most of the cell free expression based protein microarrays, and largely it can be applied for all type of protein microarray platforms, but it will be more specific in the context of NAPPA. So, first of all, we need a repository of the expression ready clones, which should be in the flexible cloning system. We have talked in the previous lecture about recombinational cloning, which provides very high quality master clones. Now, those master clones can be easily transferred to the subcloning different type of expression systems. So, if you have different vectors, you can transfer those from those master clones. Then you need a pipeline for very quick purification of DNA constructs and how to array or print them on the chip surface. You need robust printing chemistry, as well as, since you are aiming for the high throughput experiments, you are aiming for thousands of feature printing on these arrays. So, you need high throughput capability at every step. So, in general, when we talk about NAPPA protein arrays in the workflow, we will talk about first of all what is the array design and the experimental planning required for performing these arrays. So, unlike other proteomic techniques where one starts with unknown things, and also after doing lot of experiment, then only they can identify the proteins by using mass spectrometry. In the protein microarrays, people are already aware what type of contents are printed on the chip surface to start with, and those contents has to be very well planned. If you have a specific objective in your mind, you want to study protein-protein interactions, you need to have some good positive controls for the protein interactors as well as some negative controls. You want to identify some biomarkers. For example, you are looking for serum screening. You need to have again some good quality control spots such as human IgGs or mouse IgGs. So, lot of thought process goes into the designing the experiment for the microarrays, as opposed to what one do in the other type of proteomic techniques. Then what type of surface one need to select, what printing chemistry need to be used. And since we are talking here about the cell free expression based system, you need the DNA to start with. So, how to express those and construct those clones. Now, once you have done the culturing, then you need to purify and concentrate DNA. Almost all the molecular biology labs perform these experiments on day-to-day routine, and it is very straightforward to do the DNA extraction, DNA concentration, etc. But when we talk about doing the experiments in the high throughput, then it poses several challenges, because the quality of all your features has to be very good. So, various type of quality control checks, various type of observations one need to keep in mind for doing the simple experiments like even DNA purification and concentration. Then once your DNA is prepared, then you need to mix that with the proper printing mix. In this case, we are talking about NAPPA arrays. So, we need to have those components which are required to make the proteins by using the cDNA. Now, array printing, how to ensure that printing quality is good, and then different type of quality control checks are required. So, just an overview, one need to do the experimental planning, how to design those arrays, what type of all the features are going to be printed on the array surface. One need to design these experiments very thoughtfully. What is the features going to be printed on these arrays? What type of chemistry need to be selected? how to purify the DNA. And once you have the slide which is functionalized, that is ready, you have the DNA in the printing mix, then all you need to do, print those on the large number of slides, and you have chips ready for various applications. So, let us start with the array design first of all. As I mentioned, a lot of thought process goes even before starting these experiments. One need to do literature review, one need to look for their experimental objective very carefully even before moving forward for the any of the printing steps. So, design experiment type and appropriate target protein test set. Once your test set works fine, then one can actually expand that for the large number of features. Now, fit NAPPA chemistry utilized to assay being executed, decide what type of array density you are planning to use and how you are going to map that. Then what are the robotic parameters one need to use for doing these experiments? So, all these steps have to be very well planned in advance. So, for example, I have shown you few insets here. The very top panel in the slide shows different genes which you would like to make on the 96 well plate while doing the DNA prepping. So, first of all you have the clones and now you want to do the DNA prepping. You know in each well, for example, A1 what gene is going to be there, for example, BCL2. Then in the H12 you have left it blank. Now, G12 similarly, like you have all the wells, defined genes are inserted there. Now, each one of these plate is only giving you 96 features. You need to have multiple blocks of 96 well plates. Then you need to transfer those into the 384 well plates. So, that 4 of these plates can be transferred to one 384 well plate. And now, that 384 well plate and multiple 384 well plates are used for doing this printing. So, now on the right hand side I have shown you the array map which shows just a very high density map how different type of features can be printed on the chip. All these things have to be designed in fact prior to doing any experiment. Now, each spot need to be printed in the duplicates. Also, you need to ensure that your control features are spread throughout the array surface. For example, if you see the bottom panel, ones which are highlighted in the red, those are showing the GST proteins. Then in the green, it is showing the non-spots. So, these spots are printed in the duplicate and they are spread on the array surface, not in the adjacent to each other, so that you can look for the duplicate results, how close they are. So, just an expanded view here of a 6 by 6 spots per block. So, these are the small blocks and obviously, on the big array you have several features. Yellow one shows the genes and green one shows the control features. So, for example, here one can use 4 different type of control features: non-spot, no DNA control, a mouse IgG, GST protein, human IgG, EBNA, etc. As I mentioned, it all depends on the objective of your experiment and then suitable controls can be incorporated in the array map. So, let us talk about cloning and plasmid preparation. In the last lecture, we talked about recombinational cloning. So, researchers have started to create such collection of cDNAs by using recombinational cloning, which allows for the rapid transfer of DNA fragments from one vector to another in frame and without any mutation. Now, I have shown you earlier that how these clones are stored, these master clones which are almost similar to the gold quality. Now, those are collected in these repositories. We have several good repositories including at Harvard Institute of Proteomics, at Arizona University, MCG clones, and there are some other commercial repositories as well. So, these clones are sequence verified, they are in the usually gateway or creator vectors, which can be transferred to the suitable vectors very easily. So, one need to do the cloning and DNA extraction before performing the printing steps. So, first of all, one need to do the bioinformatic selections of target cDNAs. If you are focusing on a particular cancer or a particular disease, obviously you have to think of what are all the genes which could be involved in that disease, as well as different type of control features. So, one need to do lot of literature search and bioinformatics analysis prior to looking for what type of genes need to be printed on the chip surface. Then once you have selected the genes of interest, you need to design the primer and use the PCR to amplify those clones. Gel purify the right side bands and then move into the entry construct. Then you need to transform that in Escherichia coli, select the right transformant colony, and then obviously you would like to use those colonies for the long term. So, it is good that if you can make the glycerol stock of the same and then you store it for the long purpose. So, the DNA preparation process is very similar to what everybody performs in the regular molecular biology lab. Only difference here is that things and steps have to be automated and has to be in the more high throughput manner, because if you are doing in the tubes, you are doing 5 or 10 genes, you cannot achieve the 20,000 density. So, all these steps need to be automated. For example, I have shown on the left slide the inoculated liquid cultures on the plate, LB-Agar plate, and then those can be used for inoculating large number of blocks for doing the DNA extraction. Now, all these steps need to be modified in such a way that you have less manual intervention. If you use lot of pipetting yourself, there is a good possibility of introducing various type of errors. So, a lot of these steps are automated by using different type of liquid handling system. So, you can transfer the proper buffers, buffer solutions 1, 2, 3 by using different type of liquid handling system and robotic platforms. Just an image here to show that different type of robotic arrays and platforms can be used for performing these experiments, so that you can do the prepping in 96 well plate format. So, lysate can be loaded directly onto the DNA binding plates by using these type of robots. I will explain you the cloning as well as plasmid preparation in the high throughput manner by showing you this animation. Automated clone storage system. In order to program the NAPPA arrays, first of all we will require access to the cloned cDNAs representing the target proteins. The cDNA clones are stored in the repositories in -80 degrees. All the genes should be fully sequenced, the full length open reading frames captured into a recombinational based cloning system. Genes of interest can be easily transferred into any expression vector by simply adding the appropriate recombinational enzymes. This process is highly efficient and enables the transfer of many genes simultaneously into different expression vectors in a single step, in frame and without mutations. By using recombinational cloning approach, the coding sequences can be transferred into any expression vector in simple overnight reaction. After the cloning is done, perform the spotting of the glycerol stalk on the LB-Agar plate. So, transfer the bacteria from the glycerol stalks to an LB-Agar plate that contains appropriate antibiotic. You can use that either using multi-channel pipetters or using liquid handling systems. It is preferred to do this in 96 well plate format, so that you can avoid any mislabeling of the samples. So, allow the bacteria to grow overnight at 37 degrees before inoculating that into the liquid medium. So, as shown here, you can use a multi-channel pipetter or you can also use the liquid handling system. So, first of all, you need to prepare the culture blocks and add the liquid broth in 96 well plate format. You need to ensure that you have added the antibiotic solution in each well. Once culture blocks are ready, then inoculate the liquid culture from the bacteria grown on the agar plates. Similarly, you can fill all the wells. Now, then cover the deep well block with a permeable seal. Ensure that sealing is proper and then incubate the cultures on a shaker. You can grow the culture at 37 degrees at 300 rpm. Ensure that shaking is uniform. After these blocks have completely grown the bacteria, for the overnight or 10 to 12 hours of incubation, then we need to pellet down these blocks. So, you can centrifuge these blocks for 15 minutes at 2000 to 2500 rpm. While doing centrifugation, ensure that centrifuge is balanced. So, once the centrifugation is complete, then the bacterial pellet will be in the well and then you need to remove the supernatant. So, remove the supernatant from these bacterial cultures. Discard the tips, and once you have completed that for all the wells, then you are ready to perform the DNA plasmid isolation. So, remember your basic molecular biology and you need to prepare the solution 1, 2 and 3 for doing the plasmid preparation. So, add 200 microliter of solution 1, which is resuspension buffer that contains 50 millimolar of tris at pH 8, 10 millimolar of EDTA and 0.1 mg per ml of ribonuclease. So, you need to add the resuspension buffer around 200 microliter to each well. So, remember in the last step we had made the bacterial pellet from those culture blocks and now you can add this 200 microliter of resuspension buffer in each well. After adding the solution, then you can close the plate by adding a cover slip. Once the seal is placed, then you need to vortex it. One can also do it by pipetting up and down or doing the vortexing few times. In similar way, add 200 microliter of solution 2 to each well. Solution 2 contains 0.2 normal NaOH and 1% of sodium rhodosyl sulphate. This is a lysis solution. After adding this solution, again you need to repeat the same process of adding 200 microliter of solution 2 to each well, seal the plate and then invert the plate few times. Once solution 2 is added, make sure that you invert the plate few times. Now, add 200 microliter of solution 3 to each well, similar to what we have seen earlier. The solution 3 is the neutralization solution, which contains 3 molar of potassium acetate at pH 5.5. Now, centrifuge the block at maximum speed for 20 minutes at the room temperature. Once centrifugation is complete, then you need to discard these supernatant by dumping the block over a waste container. So, once you are ready with the clones, you have the good DNA preparation. Now, you need to make the slides, you need to functionalize the glass slide surface for printing purpose. So, we will talk about Aminosilane coating here. So, one can use different type of surface, for example, glass, gold, nitrocellulose, hydrogels, etc. There are different type of properties one need to look for while selecting the right surface. Obviously, cost is one of the major factor when you are talking about experiments such as biomarker discovery. You need to use hundreds of chips for screening hundreds of patients. So, if you have glass slide which is of low cost, then that will be more affordable for large experiments. So, gold is obviously very high cost, so as nitrocellulose and hydrogels. The reactivity for the glass is moderate, so as the nitrocellulose and hydrogels, but gold has low reactivity. Absorption to surface, that is low in the case of glass and gold, it is high in case of nitrocellulose and hydrogels. Now, compatibility for the mass spectrometry, that is only applicable in case of gold surface. Now, these surfaces can be derivatized by using different type of chemistries, including amino groups or thiol ester. So, we will focus more on the silane based chemistry. We want to talk about printing the DNA on the chip surface for using cell-free expression based NAPPA protein microarrays. So, silicon based chemicals, they contain two types of group, the inorganic groups such as alkoxy group like methoxy or ethoxy, or organofunctional groups such as amino, epoxy, vinyl, etc. So, the DNA can be attached covalently on the glass surface by baking or UV irradiations. One can bake the arrayed site at 85 degrees. Also, UV cross linking is a popular method which also provides good result for DNA mobilization. So, aminosilane coated slides, they provide high concentration of primary amino groups available at the chip surface. Now, why we need to use this chemistry? So, amino group provides a positive charge, and how that will help? When placed in the contact with the neutral aqueous solution, the groups becomes positive charge. Now, DNA backbone that provides the negative charge. So, DNA backbone will form the multiple ionic interactions, as you can see in the slide, with positive charged amino surface coating. For performing aminosilane coating in the lab, one need to have the glass slides, acetone, aminosilane, usually 2% aminosilane solution in acetone works best. Then you need metal slide racks and you need a rocking shaker. In this slide, I have shown you various steps involved in performing aminosilane coating. On the left side, a rocking shaker is there, in which a glass tray contains metal rack containing 30 slides. On the right side, on the insert, you can see those 30 slides are placed in the metal rack, which is submerged in the aminosilane solution. On the bottom panel of the right side, I have shown that these slides can be either centrifuged or dried with the compressed air, and then those are ready for further printing purpose. So, let me show you this animation how to do the aminosilane coating. Prepare 300 ml of aminosilane coating solution, which is 2% of aminosilane reagent in acetone. Place the slides in metal rack. Treat glass slides in aminosilane coating solution for around 15 minutes in glass box on shaker. Rinse with acetone and followed by brief rinsing with milli-Q water. After washing steps are done, then you can spin dry in the speed bag. So, now let us talk about printing arrays. You have DNA ready, as well as you have the glass slide functionalized, that is also ready. So, now you need to think about how to print those features on these chip surface. So, first of all, you need to make a master mix or printing mix, and in case of NAPPA, as I mentioned earlier, you have 4 features: BSA, BS3, the cDNA containing the GST tag, as well as an antibody which is anti-GST. So, let me briefly explain you the role of each of these 4 components. So, BSA dramatically improves the DNA binding efficiency. BS3, BSA and capture antibody, they are coupled to the amine coated glass surface via the activated ester terminated homobifunctional crosslinker. The cDNA contains GST tag, or you can introduce any other tag. Then you need to have the capture antibody, so that the expressed proteins can be captured with the antibody to a C-terminal GST tag on each protein. Once we have these 4 components ready, then you are ready to perform the printing. So, what is the role of BS3 crosslinker? It is water soluble, non-cleavable and membrane impermeable. The amino-reactive groups of N-hydroxy sulfonosuccinamide sulfo-NHS are separated by the spacer of 8 carbon atoms, and each protein contains a terminal amino group. Now, once you have these components ready, you are ready to perform the printing. So, on the softwares, you first need to define various type of parameters. You need to define what type of pins you are going to use. For example, one can use even 48 pins for printing several features simultaneously, or even go as low as 1 or 4 pins. This could be of different type of microns, like 300 microns, 150 microns, depending on what type of density you want to achieve. You definitely need to ensure that there is good humidity maintained throughout the experiment. So, at least 60% humidity should be there. You need to define the source plate which you are going to use for printing, whether you have one plate or you have multiple plates, and what type of arraying pattern these 48 pins have to follow. All those things have to be written in the programs on the software, and then only once you place your plates, 384 well plates containing the master mix, those can be used for printing purpose. Now, one need to ensure different type of robotic parameters are in place. For example, environmental conditions such as humidity, temperature, vacuum check, all these things need to be ensured before you place your plates for printing. The pin size and the number of pins, again depending on the density which you want to achieve. How you want to set up the deck for printing, you want to use the plates, you want to use the slides, how many number of slides you want to use. Again it depends on different type of arrays which are commercially available. One can use large number of slides. Array mapping, that is already you have planned experimentally beforehand, what density you want to achieve, where each of your genes are going to be located on the map. Now, what are different type of printing parameters such as inking, stamping, print depth, and then you need to ensure that in between the printing the washing is sufficient. So, how the pin washing is performed, and you definitely like to track and log all of this detail, so that if during a large printing anything goes wrong, you can actually troubleshoot and know that at which step things have gone wrong. So, printing good quality arrays is very challenging, but once you have the good quality arrays in your hand, then you can perform different type of experiments. So, let me show you this animation to explain you the procedure for master mix preparation as well as printing procedure. So, prepare enough master mix so that you can do printing for large number of arrays. The master mix contain DNA, polyclonal GST antibody, BSA and BS3 linker. For detailed recipe of each of these reagent, you can follow the publication manuscript by Ramachandran et al. 2008, published in Nature Methods. Once master mix is prepared, you can mix it well and then transfer 20 microliter of master mix to the block containing plasmid DNA. Once the master mix is added to all the wells in the block, they need to shake the plate for 15 minutes. For printing, you will require 384 well plates. So, now you need to transfer your master mix containing plasmid DNA from the 96 well plate to the 384 well plate. Again, these steps can be performed either using multi-channel pipetters or by using liquid handling systems. Once master mix is prepared and transferred into the 384 well plate, we can perform the printing step. So, we can use these microarrays to print the DNA and the master mix on aminosilane coated glass slides. First, you need to ensure that pins are washed thoroughly. You can wash with ethanol and water, and during entire printing procedure, we need to ensure that humidity is maintained at 45 to 60 degrees. One can use different type of pinheads and different type of microarrays for printing the chip. Now, these master mix can be printed on the chip. So, what are different type of control features you need to put on the array? Let us have a brief discussion on that. So, as briefly mentioned earlier, you need to ensure that different type of control features are spread across on the array surface, and these could be mouse IgG, no DNA, non-spot, EBNA, human IgGs, etc. Now, to test the background, you need to have the certain control features. Now, since we are talking about NAPPA arrays, we are talking about the clones containing the GST tag. So, it will be a good idea to have the different type of spots with the GST alone. So, you can have the purified GST in the dilution series, so that you can use that for doing the calculation for protein expression later on. Now, if you are looking for different type of immunological response, biomarker discovery, etc., you need to ensure that IgG is printed at the varying concentration to assess the immunological responses, such as I mentioned for screening human biomarkers. It can also use for the statistical analysis later on by the biostatistician, so that different type of protein expression can be normalized. So, another control feature and the QC check here shown on the slide, same gene printed 4 times in the blocks repeated, and then negative controls are used to give an idea for the background. Now, what are the different qualities one need to keep in mind for good protein arrays? Obviously, you like to know the spot size, spot shape and morphology. Those have to be uniform. So, spot to spot consistency has to be there, positional consistency has to be there for the large number of slides, sample loading, sample integrity, as well as different type of controls, replicates and positional flexibility. All those need to in place for good quality protein arrays. So, just shown here, one can actually encounter different type of problems while printing these arrays. So, shown on the left is spot size, shape and morphology. Shown on the right is spot to spot consistency, and many times these type of printing issues do come while performing these high throughput experiments. So, one need to ensure that all these parameters are properly quality control checked. Again, like looking at the spot morphology and the uniformity of the spot, one can think of various type of parameters which could be useful. For example, spotting solutions, spotter capability, maintaining the temperature, maintaining the humidity conditions, keeping the dust free environments, etc. Now, printing the quality arrays, that is very useful. You need to have the precise liquid handling systems. As I mentioned, that each step can be automated for the same. You need to do the very strict tracking of the entire process, so that you can troubleshoot if anything goes wrong. It is a good idea to build a detailed log history for each step, starting from the cloning, DNA preparation, printing, etc. Now, if you have done this step by step optimization and evaluation, there is less chances of making any errors, because in high throughput approaches, when we are talking about thousands of features, it becomes very essential that everything has to be tracked throughout and logged in detail in the logbooks. So, now let us talk about, we have done the printing, we have ensured that our printing has very good quality. So, now can we use these NAPPA arrays for doing the further biological experiments? So, first of all, since we have printed the DNA, we need to ensure that DNA printing is good. So, to assess that, one can use the picogreen staining, and next, once the DNA quality is good, then one need to ensure that protein can be expressed from those DNA by using in vitro transcription and translation system. So, the protein expression need to be quality checked. So, as shown in the slide, a very small test array which I used to teach in the Cold Spring Harbor to the students, and these are actually made by those students. So, in the bottom you have a map where only 5 genes are there: CDK2, CDK4, June, P53 and P21. Each one of those genes are printed in the duplicate. Then you have master mix and you have water. A very simple proof of concept test array. Now, only 5 features, 5 genes should have the DNA. So, if you do the picogreen staining to observe the DNA quality, then as you can see on the upper panel, those green spots are lighting up, showing that DNA is printed properly. One can use the different type of statistical tool for analyzing how good the DNA is printed on the chip surface, which is shown on the graph in the right side. Now, different batch to batch variation need to be ensured, because if you are printing 200 slides, 100 in each batch, you need to ensure that all the batches can be used for one experiment and those data can be compared. So, always printing batch to batch should be ensured for both DNA staining as well as the protein expression. Now, once the DNA is quality control checked, then we need to move on to the protein expression testing. This is the schematic showing that what are different steps involved in performing NAPPA based protein expression. So, you have the slide, you need to block those, then use the in vitro transcription translation system, so that proteins are expressed. And now, you want to detect the signal. So, you can add the primary antibody, anti-GST in this case, and then followed by the secondary antibody containing either Cy3 or Cy5 labels, or you can use the HRP based tyramide signal amplification system, TSA based systems. So, shown here is again a proof of concept array map with only 5 genes, and these proteins need to be QC'd here. So, one can use the anti-GST antibody, and as you can see, only 5 proteins in duplicates are expressing, whereas master mix and water are blank, and same can be plotted on the graph which is shown on the right hand side. Now, you have assessed that you have good protein expression, but how specific that protein expression is. To ensure that protein specific expression, one can use the specific antibodies. For example, in this case we have P53 also on the chip surface. So, we can use anti-P53 antibody, and as you can see in the middle panel, only one pair of the spots are lighting up, which shows that only P53 protein can be detected if you use the anti-P53 antibody. On the right side in the graph, only one spot is showing the graph bar. So, it shows that in a high density array, similar type of quality control checks can be performed for the protein expression shown in the left side, P53 expression or the protein specific antibodies in the middle panel, and the inset showing that how duplicate spots can be looked at. So, those duplicate spots are for the P53 proteins. This is an overview of the different type of quality control checks which one need to perform during the whole NAPPA step. Before you use these slides for doing any biological questions, you need to ensure that DNA is properly printed, shown in the left side in the slide, picogreen staining. Next to that is the protein expression by using the anti-GST antibody. Next is the specific protein expression, for example, using anti-P53 antibody, and then looking at the inset for the those duplicate spots. Again, one need to look for the batch to batch variation after the protein expression, and if you print the GST protein in the dilution series as shown in the bottom side, which is printed in the bottom of the slide, those can be used for assessing how much protein is produced on each feature. So, batch to batch CV, sample to sample coefficient of variation, as well as replicate coefficient of variation should be calculated. Now, once you have looked at day to day variations, then also look at different type of other variations such as spot to spot variation, slide to slide variation, and day to day variation. Again plotted in these in this graph. it shows that NAPPA arrays are quite reproducible and the R square is more than 0.95 in all the cases. So, let me show you this animation for the quality control check, how to detect the DNA and the protein expression on these chip surface. Once the arrays are printed, then we need to perform the quality control checks whether DNA printing was appropriate on these chips. The storage form of NAPPA microarrays is merely DNA. So, in an activated array, all following reactions elapse in the solution and in the real time. Therefore, restrictions caused by the instability of proteins occur very rarely. To perform the DNA staining, first prepare the pico-green stock solution, dilute it in the milk or super block. Block the chips with the super block for an hour, and after blocking is done, then we can use the pico green mix to place on these printed slides. So, let us first look at the NAPPA expression on the slide, and then I will describe about scanning for both DNA printing and the protein expression at the same time. So, first you need to prepare the in vitro transcription translation mix, which contains TNT buffer, T7 polymerase, amino acid, RNase inhibitor and DPC water. Apply that on the slide, which contains the Hy-Breville gasket, and then seal the pores. Now, this printed slide which contain plasmid DNA, they have the in vitro transcription translation mix shown in the red color here, and the next step will be to incubate these slides for the protein expression step. So, incubate the chips for 1.5 hour at 30 degrees for protein expression, followed by 30 minutes at 15 degrees, which allows the protein to bind on the anti-capture antibody. After this incubation is done, then we need to wash the slides. So, remove the Hy-Breville, wash with milk in PBS for 3 times, 3 minutes each. Now, block the slide with milk or super block at the room temperature for an hour. After blocking is done, then we need to drain the excess liquid on a paper towel, and then we are ready to perform the incubation with the primary antibody. So, apply primary antibody, which is mouse anti-GST in this case, because we are looking for the protein expression and all the clones contain a GST tag. So, we need to add the primary antibody on the chip, which is already expressed proteins are there, and after adding the primary antibody, we need to incubate it for an hour at the room temperature. Now, place the cover slip so that liquid is uniformly placed, and then we need to perform the washing steps after an hour. Proper washing is very important for all the microarray experiments. Now, we need to add the secondary antibody, in this case anti-mouse HRP, and incubate again for an hour at the room temperature. After secondary antibody incubation, we need to wash with PBS 3 times and then apply Tyramide signal amplification system for signal detection. After adding the TSA solution and incubation, then we need to wash the arrays in water very quickly, so that excess TSA is removed. Now, we need to dry the slides with the compressed air, or drying step can also be performed by using centrifugation. Now, we will see animation for the scanning slides. Whether you have done the QC experiment of DNA detection or protein expression, now you need to scan the slides. The different type of scanners available, including those ones which are automated, and multi slides can be scanned in the automated way. First, you can preview the scan, and then you can adjust different type of settings to see the signals better. Then you can look at each region in the zoomed in view and look for the signal. So, in summary, today we discussed about how to make NAPPA protein arrays. Similar type of concept can be applied for other cell-free expression based systems as well. To make the NAPPA arrays, one need to have the access for cDNA clones. One can do the recombinational cloning or obtain the clones from various repositories. Then grow those cultures and make the good plasmid preparation. After that, add the master mix containing BSA, BS3, anti-GST antibody, as well as cDNA containing the GST tag. Transfer these master mix from the 96 well plate to the 384 well plates, which can be used for preparing the array plates. Now, these can be used on the microarrays, which can use the printing. Once printed, these slides can be stored on the room temperature, and then one need to use the different type of quality control checks, such as picogreen staining for DNA detection, as well as adding the in vitro transcription translation mix for doing the protein expression. Once these chips are ready and one has performed all the quality control checks, then these arrays can be used for different type of biological applications. So, in summary, today we talked about how to make NAPPA protein arrays, but same concept also gives you an overview of various type of intricacies involved in making any type of protein microarrays. Thank you.